Shanghai Jiaotong University, and he's a associate professor there. And from Guomix at Westlake University, we have Dr. Zhiwen Liu, who's a postdoc researcher, and we have Mengge Lü, who is a PhD student. Last but not the least, we're honored to have Dr. Tiannan Guo, PI from Westlake University. Without further ado, Dr. Morris Huso, if you would like to start sharing your screen and your talk.
All right, let's do this. Thanks a lot for the invitation.
Yes.
I'm happy to share a bit the work in Zurich that now is already a few years old, but has sparked some interesting developments, and also some work in Tiannan's lab at Westlake. So I think that's really the reason why we're here, to look into where it started and what could happen in the future based on this. Now, let me see if I can get that share going. Do you see my slides?
Yes.
Now, it would be great if I had the presenter view. I don't, but then excuse me if I need to rethink, but that's good. All right, so today I will talk to you about an approach developed about 4 or 5 years ago, or maybe it's already 6, at ETH Zurich, based on prior work by numerous research groups, and we made it a targeted approach and kind of ported the targeted proteomics, DIA targeted analysis approach onto this problem of protein complex analysis. And also what I will touch on is how then such an experiment, which is technically giving you a lot of insight in a single experiment, but still every experiment being 80 or 3 times 80 MS runs, costs a lot of money and costs a lot of time. So I'm also going to touch on how this can be sped up, and how the EvoSep1 system and EvoSep chromatography, disclaimer, my new employer, is useful to speed this up, and touch on work done by Ben Collins and colleagues in that direction. But I will not get into that too deep, but rather take a few minutes to introduce the EvoSep1 system and what it does and how it can help make scientists' life easier around the mass spec lab and enable high throughput studies to unlock new insights in biomedicine. All right, so let's get into this. Here's a short overview how I structured the talk. So first of all, I'm going to give an introduction into proteome organizational structure and how this can be analyzed by mass spec. What are the approaches that were out there, and what was our starting point, and what were our considerations really? And then go into the SEC SWOT-MS workflow, targeted protein complex profiling, and then how we also use this, or based on this workflow, managed to track rearrangements and changes in protein complexes, which is then where it gets really interesting when we start seeing this system and how the proteins are really organized, move over time, and understand more intricately how it actually all strings together and works. So dynamics, awesome, but of course then difficult given the throughput limitations. And then I'm going to touch on how this can be sped up by the EvoSep1 system, which arguably is a robust, well-standardized LC platform that makes life easier and labs more productive. And then we have the panel discussion. Okay, so let's get into the intro. I think just as a very top level view, I would just like to outline, let's say, the position of the proteome in a cellular system and in a living cell, where really it gets produced by DNA and RNA according to, let's say, these very classic single directional view, basic laws of biochemistry. But really then it interacts with a lot of different things, also with RNA, and all these biomolecules together form a network where arguably the proteins are the enzymatically active and most functionally relevant layer of that network, we proteomics people would argue. So it's really interesting how they interact with the other molecules, but mostly with one another, and really looking into that structure is super interesting. Now, the proteome is a tricky thing to study, and here on the left is just a colorful image, some eye candy for those who like it, and on the right we have some actual data. What you're looking at here is an estimation of the copy number range, showing the copy number, estimated copy number of given proteins, and you see that it ranges over about 6 or 8 orders of magnitude here, but I think by now we know it's even more, so meaning that if there's one copy of the lowest abundant, there can be as many as 100 million or even more copies of the most abundant one. And since in the protein world we don't have an amplification strategy like a PCR, it's tricky to analyze, especially the lower abundant ones, and get a complete picture of what's going on. And to make matters worse, we also have other factors like alternative splicing, post-translational modification, and other factors affecting the protein shape and function. And then in the end, we have way more protein forms than genes, and life gets hard. All right, and now to study this, I think mass spectrometry has really evolved as one of the main tools and is really powerful to do that. And out of the various mass spec workflows, here is one, which is termed bottom-up proteomics, where we digest the proteins into peptides, fractionate them by hydrophobicity and reversed phase chromatography, and that's where I will later touch on how this is done differently on the EvoSep relative to classical systems, let's say. And then this gets ionized and fed into the mass spectrometer, and we have different options how to operate the mass spectrometer. In short, DDAMS, very classical, selects one by one these precursors as they are still intact for fragmentation and records fragmentation patterns. Samples frequently in the MS1 scale space on the intact precursors, but not as much on the MS2 space. And that can then be searched with classical spectrum-centric search tools, which gives us an idea about specific peptides, where they elute and how they fragment. Now in SMRM, single or multiple reaction monitoring, we direct the mass spectrometer to a select set of peptides and continuously scan and fragment and record on the MS2 level the fragment ions in order to get a very selective and specific and very high quality quantification of this particular peptide, for example, for the validation of biomarkers in large cohorts. And last but not least, developed over the last, let's say, 15 years is DIA mass spectrometry, SWOF-MS being one incarnation of that, where we now remove most of the stochastic or targeted analysis steps and just subdivide the signal space into several bins and just cycle over that over and over again while co-fragmenting the multiple precursors contained therein and recording very high density multiplex MS2 spectra as well as MS1 spectra, and all at a high sampling rate, generating a very dense and deep record of the peptide set eluting from the column. And that really now is a cool tool, I would say, and was what I found in the Ebersol lab back in the day to work with. So that was really neat to have such a, at that time, new technique available to try and do the bioanalytics, especially of protein complexes, in a different way. So now let's dive into how these are analyzed. We have the MS2 spectra generated from the DIA method. And classically, we would have used prior evidence and the information of the DDA searched results formulated into a spectral library, prior information on where we would expect this peptide to elute in the retention time space, precursor MZ, fragment MZ, and thereby we can, in this dense data, look for and extract specifically a signal for that peptide, which reflects closely the signal and the quality one would have gotten from a targeted SMRM experiment. And based on that, arguably one can achieve deep proteome coverage. And this is visualized on the right here, where this cylinder represents, let's say, the proteome space ranked by abundance, abundant ones on the top, lower abundant on the bottom. and this, let's say, golden cone, the coverage, what can be seen by the mass spectrometer. And whereas in DDA, there's broad coverage at high abundance due to this semi-stochastic precursor selection step and instrument scan speed limitations, we quickly lose coverage as we go to lower abundance. And in SMRM, we can go very deep in terms of abundance range, but really only on a very limited set of peptides. And there are still limitations here when it comes to the instrument scan range and dynamic range of scannability. But now in DIA, we argued, and I think by now it has been proven, that it is really a nice merger of the two, where you can ensure breadth, but also get quite deep and don't lose breadth as quickly as you do in a classical DDA measurement in the same gradient of the same sample. So that's a cool tool to work with. Now, here I just visualized or collected some nice visualizations of how this works. So just to get an idea. So let's look at the lower left one here. We see the time axis and the different MS2 scans that have been collected. And now each individual fragment ion produced by a peptide eluting off the column can be monitored and followed. And this is this peak group, which really is the evidence, and the co-elution of these three specific fragment ions is the evidence for this specific peptide. So that's neat. Now, how does this look in practical terms? My favorite example here, you see the same sample shot and left in DDA, right in DIA, and a M/C over retention time heat map. And now I'm going to visualize the MS2 events. On the left, you see each individual precursor that the machine had time to look at. And on the right, we see the center of the MS2 scans of the DIA method. And I add here the range of the scans. So everything within the red boxes on the right has actually been sampled. And on the left, only the things that are marked by the black dots are sampled. And I hope you agree that some are not sampled. And that's a picture from the SciEx people, how they like to represent it. Really, there has been a complete harvesting of what should be in there and visible to the mass spec. Okay, this is a representation of the same thing. On top DDAMS and on the bottom DIAMS. And what I want to highlight by this is really the concept of having a spectral library and prior knowledge, which is high quality, but let's say more sparse. So it has been acquired one by one, and it might not cover the whole space. And we have a broad record below based on the DIA method. And by merging them together in the targeted peptide-centric analysis, that's how the performance of SWOT-MS could firstly be demonstrated and yeah, be shown to be pretty good. Now, talking about proteome organization and what we are looking into, this is just textbook knowledge extended a little bit from left to right. We have the primary structure of sequence, then the tertiary structure, how it folds up, followed by quaternary structure, which then I would say is higher order organization. Could be homo oligomers like homotetramer, or we can then go into the hetero-oligomers, protein-protein complexes, as we typically think of them, where different proteins interact to then carry out a certain function or multiple certain functions together. And we can also think about network structure when we think over a time axis and maybe some of these complexes not being stable, but rather, signaling complexes and so on, that this is another structural layer, especially over the time axis and changing. Okay. Now, I would like to show one example, and an example here is that classically we thought one gene generates one protein and that carries out one function. But actually, it's a bit more complicated than that, because we have multiple genes, they produce multiple proteins, and we're neglecting other things here. And you realize that, oh, protein A does not execute that function, but only once it has its partners available, it does execute that function. So really showing that it is critical to some proteins' function that they are assembled in the correct way, and this higher order assembly is crucial for how our cellular systems are regulated. Now, a real-world example for that is the COP9 signalosome complex, which denatylates CSN, which denatylates E3 ligases via its catalytically active subunit CSN5. And only when CSN5 is integrated in this octameric complex, it really does this. So if you take it in isolation, you won't get the function. And that is a very important complex. And if this wasn't the case, then we all wouldn't be here, basically. All right, so which techniques are there to study the proteome? There's classical techniques based on, let's say, functional complementation, where you split a protein in two and add reporter ions like yeast to hybrid, and then you put it into yeast or into the other organisms and get a gene readout, whether they did interact or not. So you fuse the two partners to two complementary portions of the reporter. And that's, of course, very tedious, but amazing studies have been done using this technique, and it really helped a lot. In addition, there's, let's say, affinity reagent-based approaches where you need a specific antibody for one or two out of the components to really pull them down and see if they co-isolate in a Western blot. Also tedious, but also powerful for those that are amenable to these methods. And a little bit higher throughput, I would say, was then when mass spectrometry kicked in as a technique and ATMS became a thing, and one figured out how to deal with the data. And I think here the Bioplex network and Ed Hutlin's work were just very impressive, assembling these data from pull downs. So you have affinity tagged proteins that you introduce into a cell, and you just pull them down and see who comes down with them. And from that, you can generate this in-depth knowledge of, let's say, individual proteins, interactome vicinity, always, of course, biased a bit by the experimental parameters and what is stable under the washing conditions that you apply. Now, last but not least, comes the basis for the work that we're talking about now, and that is protein correlation profiling mass spectrometry, also termed co-fractionation mass spectrometry, which is relatively new. I think since 2008 or 9, the first papers have been coming out working in that direction. And the idea here is that you basically have a soup of complexes and non-complexes in your cell, and you can technically isolate them and keep them intact as much as possible. And then if you fractionate them out by different physical-chemical properties and the complexes are intact, they will co-migrate in the respective chromatographic dimension. And then by having a quantitative readout by the mass spec, one can then calculate back and calculate correlation coefficients to infer and estimate which out of those are likely interacting with one another. And as a result, you get these pairwise interaction-centric scoring maps, which can then be clustered into something that one would think is a complex. And that's what has been done there. There's really important work in that direction. Just very briefly here, also to give fair credit to the work this was based up on. I think what came first was subcellular localization profiling with density gradient centrifugation, where proteins from different organelles locate with these organelles differently along density gradients, and there was important work done here. And then when clustering these data, what people actually saw was one cluster that basically only contained proteasome proteins. And that's what I think then really sparked those ideas. At least that's my best current theory, to say, hey, if we see the proteasome in such a density gradient centrifugation map, why don't we do more fine-grained chromatography that aims to separate the different complex versions that are there, and then ion exchange chromatography in combination with density gradients and other dimensions, also SEC, kicked in these early studies where people tried this out and achieved amazing results. And when we started this work, the status was about 2012. There was this landmark study, really beautiful, from the Foster Lab out of British Columbia, and Anders Christensen, who did a tremendous job there with SEC, and HuB et al. That's the Emily lab, who did this multi-dimensional separation and immense development to draw the first map of soluble human complexes. All right. Now, yeah, that shouldn't have been there, sorry. Now we addressed this, let's say, state-of-the-art technique, and we really sat down and thought, how can we try and improve this? Because it's really cool, but the coverage is still very low, and maybe we can do something about that. So we looked at the process, what was currently being done, and thought about what's really limiting. And what was really limiting, we thought, was one, the resolution. I'm not sure if you guys can see my mouse pointer, but let's look at this box on the left. It's the resolution on the, let me see if I can, no, okay. Right, so the resolution of the chromatographic separation would be important to just separate different complexes and avoid false co-elution of proteins that don't really interact but participate in separate complexes that happen to come out at the same time. And when maybe with a bit more resolution, that would get better. But then there's also the breadth of the analysis in general, and also the quantitative accuracy, meaning the goodness of the correlation signal between two truly interacting proteins versus two not interacting proteins that we thought could be optimized. And secondly, we also looked at this interaction-centric scoring, where I think back then people were just, let's say, squeezing the co-elution data into analysis frameworks and a thinking framework really of this APMS-centered network representation of modular biology, where they really wanted to get a score for each single interaction, and then just did it pairwise. And fair enough, that's a good approach, and we tried that also. But I would think there is certain limitations of this, and I will get into this, and that's how we came up with our complex-centric analysis approach. All right, so to address these prevailing limitations, what we did was, excuse the horrible animations. So what we did to address these on the left first is to say, well, quantitative accuracy and breadth of the analysis, we really think DIA mass spectrometry can make a dent into these two angles of, let's say, limitation. In terms of breadth, we also thought a mild detergent assisted complex extraction method, which leaves the complexes intact, but yet gets more of them out, especially membrane associated complexes, could just help with the overall coverage, and just translated a protocol that had been developed in the Eversort lab before, based on NP40 as a mild detergent, which had successfully been employed for numerous APMS studies and had shown to not disintegrate complexes, but improve recovery of complexes from cells, which we could play as a card towards this dimension of prevailing limitations. And in addition, we just looked up basically the mildest chromatography type we can think of, and that's SEC chromatography, because you never, theoretically at least, you never have a physical interaction between your analyte, the complex, and the stationary phase, the beads inside the column. And there was new materials around at the time, 3 micron Phenomenex, Yara 4000, which really added to the resolution of this technique. So that's what we went for, to just try and, give this our best shot and see what we can do. And secondly, on the right, here now is the approach that we pursued regarding the analysis of these data. And we just, looking at the data manually, we thought, if there's a trimer, that's 3 binary interactions there, but at the end of the day, it's a signal that is composed of 3 peaks, let's say, and it really looks to us like a peak group in DIA, and it is a signal that is composed of 3 and not 2 entities, and we should score it as such. All right, good. So with that, I dive into the specific implementation that we came up with, SEC SWATH-MS. So this is the workflow. Like I said, we did this mild complex extraction, detergent assisted, coming from a cell, fractionated by size, and then record this, all the proteins eluting. Well, first we record the peptides, and then from that reconstruct the proteins. And then for the analysis, we really do a targeted analysis using a protein connectivity library as prior knowledge and a target decoy model that the team developed, including many of the computational members of the team that did really a tremendous job here to make this fly and work, and put together over many iterations and versions the now called RCC profiler algorithm that assists with an error-controlled workflow to derive these signals and get a nice summary what has been detected. And that was our first paper, the MSP paper, from the title. So that was really the big outcome. And I want to illustrate, and excuse all the animations here, they somehow prevailed. I want to illustrate the extension of the targeted proteomics concept. So really down here, we are looking at the peptide-centric targeted analysis of swath MS data or DIA MS data, where a peptide ion elutes from the C18 column. And then a peak group in the C18 retention time space is the signal that qualifies and quantifies this peptide being present in our sample. And we only get there by having a good reference spectral library to look for these in the right place. That said, I guess one must say that nowadays this has been kind of replaced by excellent prediction of peptide retention times. But still, I would argue that most tools still predict the library and then use it. Okay, so this signal space in DIA is then comparable to the signal space we have coming off of the SEC column in the upper two rows here. And first, there is a, let's say, protein-centric layer where an intact protein comes off the column, and its component peptides, the proteotypic peptides, form a SEC chromatogram peak group. And for that, we don't really need prior knowledge. We have our FASTA database from Unicrot, so that's kind of luckily done already. And then for the protein complex level, we argue that if it was intact as it came off the column, then we have the peak group in the SEC dimension. And here now we do need additional prior knowledge, and we argued that we should really integrate a lot of the work that has been done before to come up with a reasonable set of complexes to look for in these data. And just looping back to the DIA world, we were just reminded of the situation where here in the upper panel we have swath acquisition and a broad record, and based on a spectral library and prior information from a more sparse acquisition method, data-dependent acquisition here below, we can interpret this, and that's really what we wanted to do for the complexes as well. So again, this comparison and analogy that we employed was that we have a broad record of complexes eluting from this SEC column along the fractions by our experiment. And the sparse information on protein connectivity came from ATMS and or other methods, right? Everything that goes into these databases we use. And then from that, we can interpret the presence of certain protein assemblies or not. And this is how this then looked in terms of steps done in the software. So we start out as input with protein correlation profiling data and also this protein connectivity information. Then we implemented a bunch of pre-filtering steps, and also importantly a target decoy model where really Isabel did a fantastic job to get a randomization but avoid having the same complex again in the decoy set. So that really helps to then have confidence in your results and have an error model and make sure that you report things that make sense and aren't just sporadic hits. And yeah, out of that comes visualizations and summary tables. So we were pretty happy once we achieved that. And just as to a little insight into what the algorithm does, and this is really only one part of the algorithm, it has been extended since. I think this is more like an early version when it was still called SEC Profiler, and Robin Hafen, a really capable student working with me on this at the time, managed to implement this in R. It was slow, but well, it did the job. And basically what we do is as we come in with a hypothesis from either of these databases, we see a number of proteins that should be part of this complex, and then we look at the set of chromatograms, and from left to right slide a window over the data and calculate these trees based on Pearson correlation, and then mark down at which range which of the members of our hypothesis actually correlate well. And then this is followed by a peak picking algorithm to make sure we are not finding super sporadic signals. And then at the end, this is scrutinized against the decoys. Yeah, let's skip over the other part. And then on the right here, you just see an example how the data can look for here, EIF3 complex, which is arguably a very nice example, a highly soluble complex that comes out in a nice Gaussian peak shape. That's not the case for all, but some of them do this, and the partitioning into a whole complex and subcomplex makes sense from a biochemical perspective. It's known that this is like a more loosely attached lid, homologous to the concept of the proteasome with its lid 19S. Okay, so in summary, what we did there is implement our workflow from left to right, fractionate, do both DDA and DIA or swath, to also compare how much we are adding by the swath acquisition rather than the DDA acquisition. And then we go into peptide quantification the conventional way. And then the new stuff we did was really this protein-centric analysis and quantifying the proteins based on the different peptide signals. And the most important layer, I would say, the complex-centric analysis, where we came up with this library building from databases and then finding complexes that way. And now a little sidetrack into the technical world, and I will speed a little bit over that. But here we quantified how much the swath layer really helped. And what we did was to shoot the same samples in swath and DDA, and then quantified swath the regular way, open swath pipeline, and the DDA data, both with spectral counting and with MS1 quantification, as in MaxQuant 1.5317 at the time. And now in this analysis here, we can see the consistency of detection. And what we mean by that is when we on the left look at the protein standard eluting of this SEC column, we see that the peak should be maybe 8 points wide, maybe even 10, if a nice protein comes off. And now for our MS data matrix, we argued that if we require increasing number of consecutive identifications, so an increasing length of continuous identification and quantification, we should understand which of these matrices is more consistent and has more consistently quantified these proteins eluting off the column. And I think the picture is quite striking that we have a stark offset of the MS2 quantified data versus the DDA spectral count and MS1. And I really think that this was one layer that also helped in the field to demonstrate that swath does a good job, and running this the mass spectrometer this way is a good idea. Secondly, there's another dimension we looked at, which is the sibling peptide correlation. So just in short, on the right here, what we're looking at here are the peptide chromatograms of one protein quantified on top by swath MS2, below in MS1 layer, and then spectral counting, and we see the quality and just visual prettiness of the peak group deteriorating. And we can calculate the correlation coefficient, Pearson correlation coefficient between each of these peptides, and then make a mean of that across the full peak group and all the peptides available. And then we can plot that for the entire experiment. And then we have the plot on the left, where on the right is the optimum, and on the left is the worst case scenario, a correlation of 0. And I think also here we could convince ourselves that we are improving the correlation signal in the data quite drastically by quantifying on the MS2 level and doing the MS as we did it. So we were pretty happy with that picture and quite excited about what that would or could mean for the depth of protein complex signal recovery that could be achieved from such a data set. Now, last but not least, the that's precisely the third layer of benchmarking we did. So we had developed our software CC profiler to look for protein complexes in a error-controlled manner, meaning that we have a number of IDs as a function of the FDR of our model. And this has been validated against tremendous amounts of manual curation by me and others in the team to make sure that our decoy model reflects what a human interpreter would do. And what we can see here is now at the same confidence level of the error model, the number of complex signals that could successfully be retrieved from the data. And what we've seen here really, it took a while until we got there, and it wasn't a road without rocks. But once I saw that, I was very happy, and I knew this is working and it was worth all the work. So we really doubled the number of retrievable signals here when doing the experiment on the same mass spec and matched gradient length. All right, so we did get deeper, which is awesome. I'll skip over that one. And then what we did as the final analysis was to integrate these different reference complex information sets, quorum, bioplex, and string database, all integrating additional layers of information. And there's arguably more one can try, or other integrative databases by now, notably UMAP, that one could use. All of that is possible. I think CC Profiler is very flexible as to its input. And then here we ran those separately and integrated the results, collapsing all the detected features, because arguably those databases are quite redundant. The well-known complexes will be in all three of them. So we needed to make sure that we're truly only reporting unique numbers. And that's what we got out here in an area proportional Venn diagram in terms of really unique signals, which is, I would say, still an impressive number. And it is also showing how much integrating these additional databases beyond quorum, and this is quorum 1.0, so to speak. At the time, there has been a big update since, so check out the new version. But that we could, in this scenario, show and see how much was added by adding string database and bioplex as separate information sets, notably each adding an additional set of error model validated signals. Okay, so now let's dive a little bit into, like, numbers are the one thing and algorithms cool, but what I really care about is biology, and what if we can find out something new about how the complexes really work. And this here is our example from before, the COP9 signalosome complex, and the signals we got for its 8 component subunits. There's markers on the right-hand side where the monomers should come out based on their size. And we get this, let's say, 3 signals, 2 of them merged, it's just a shoulder, and then one on the right. So let's dive into what they are. So the shoulder on the left, we overlaid traces of the known substrates, which is members and subunits of an E3 ligase. So we think that likely we captured here a portion of the COP9 signalosome that's currently hooked up with a E3 ligase. Of course, we couldn't validate that, but at least the profiles suggest that. Not that we tried a lot of validation, I need to note. And the second 2 signals, especially the middle one, seems to be matching in size with the holo complex. So that's good. That's the dotted line here where that would be expected with 1-to-1-to-1 stoichiometry, which luckily for this complex we know. And the other one, and that's where it gets curious, is this subcomplex of CSN 1, 3, and 8. And that reminded me of prior work where this also popped up. I did work on this complex before, and that's why I could scrutinize the data and dive into it in that level of detail. You can't do that for 500 protein complexes. That's just insane. But here, that really matched some of our prior observations, and we could then dive into understanding whether this subcomplex could really be a assembly that exists in that form also in the cell and didn't just fall off the holo complex as a result of our lysis procedure. And we did a bunch of validation experiments there, and one being that we can produce this separately and it's soluble and behaves nicely when expressed in insect cells. So I think that is an indication. It's not finite proof, but I think we're on a good track and would believe that this is possible, that this exists in a cell. And one thing said, the subunit CSN1 is the substrate or involved in substrate coordination, but there is no catalytically active CSN5 in this assembly. So if these two coexist in the cell, then the subcomplex might as well sequester and shield substrate from proteolytic processing by the catalytically active holocomplex. So that might well be a way how this process is regulated, which in turn tightly regulates the ubiquitin proteasome system. And I think everyone in cancer research knows how critical that is. Okay, so I think pretty cool deep insights that were not possible before on the COP9 signalosome. And let's dive into one other example, which is the proteasome, arguably one of the more abundant ones, but nonetheless, I think we have seen deep into particularly the subcomplexes and the biogenesis of this complex as the subunits get assembled in the cell. And I'm gonna show you here what I mean by that. So on the left is all of the subunits profiles, and we see the version with a lid is spread out over a wide retention time range. Then comes the 20S core particle, and then comes in the range between 300 and 100 kilodalton, this funky looking peak group. And when we dove into that, what I could find by diving into the literature and correlating what is known about the assembly of the complex and how it's being constructed in the cell and which chaperones are involved, I could derive the following picture. Now let's look at the lower right, which is, let's say, the conventional assembly model, where from right to left, the alpha subunits get assembled by chaperones, let's just say red and black. And then red leaves, yellow comes in, and the beta subunits in blue get assembled on top of the alpha ring, and then black goes away, and the last beta subunit comes in, beta 7. And that supposedly triggers dimerization into the 20S particle, which has 2 copies of this AB double ring. Now, when we looked into our data, what we could find is a tight correlation of these chaperones being present in the smaller assembly, black and red matches, and in the later, bigger assembly, yellow and black being present also matches quite well, and there is no signal from the red, so it seems to have left. So it looks perfectly like the established model. But now when we look into which beta subunit is where, what we could establish is that beta 7 is already present in this assembly intermediate, while other beta subunits are lacking. And if and only if we are looking at the timescale of assembly here, what might be the case is that there exists an alternative assembly route to the 20S proteasome, which depends on other interfaces and triggering mechanisms for dimerization of the 2 double rings into the 20S particle. And again, related to cancer research, if we can suppress assembly of the 20S proteasome, I think that's a pretty neat alternative to chemical inhibitors, which cause a lot of problems. All right. So let me check the time. Unfortunately, I don't have the presenter view here, and I see I need to speed up. Now we did these viewer apps because I thought, this is 2 examples where I could dive into with some background knowledge and reading to really make more sense of the data and come up with true biomedically relevant hypotheses that require further testing, but warrant further testing as well. So I thought we really need a viewer, so people with proper background biochemical knowledge can look into this data the same way, and they should be still live. And I still encourage everyone to go take a look. It's amazing what one can find and what people can find who know a certain complex and biological domain when they review these experiments. So if you ever think about running your complex over SCC column and Western blotting along the fractions, don't just check if it's in our data set. That should give you a very, very similar result, just with better quantitative accuracy in our case. Good, all right. So just as a, let's say, zooming out a bit, bird's eye perspective, what have we done? with this technique, we take a cell system, we really take it apart, we get the complexes, we break them into subunits, we get the proteins, we digest them into peptides, and we fragment these peptides into fragment ions. And then using the same approach of this peak group representing a certain analyte, we reconstruct the system and derive quantitative information as to the mass distribution and current, let's say, occupational behavior of the mass of a protein being present in its cell system. And we really believe that this is an important layer of how the cell regulates its current program. All right. So let's dive into tracking complex rearrangements. That is the second study we did, where we said, okay, now this is up and running, but we can only surmise a temporal axis in the assembly intermediates we saw from the COP9 signalosome. But let's do a real temporal measurement, and let's try and start out with something most relevant and most profound, which is the cell cycle, where with collaborators from the Institute of Biochemistry, notably Mario Köhler, and Ulrike Kutai, they did this experiment to arrest HeLa cells in two distinct cell cycle states: interphase, early S phase, and mitosis. And then from each of those, we performed the triplicate label-free experiment as we did it before, while optimizing a little bit, trying to, take a bit less fractions, be a bit fast on the mass spec, because this was about 400 runs production mode, not counting any development overhead. So this was a massive study. And what we got out is a matrix over 5,044 proteins in interphase and mitosis. And then we were faced with yet another challenge. How do we actually now score the changes that can be seen and differentiate them from sporadic changes? And the first analysis approach we came up with here, and which we used for the study, was the differential association testing, i.e. protein-centric differential analysis, meaning that in the protein-centric approach, we detect the peaks of the peptides, how they come off the column, and that's then of one protein. And we can differentially quantify between the two experimental conditions, and thereby create for the different SEC ranges a differential testing experiment, so to speak. So by subdividing the protein signal into distinct elution ranges, a regular differential expression testing algorithm can help us factor in the level from the SEC dimension. That said, of course, as you might surmise, and as the reviewers correctly spotted, that is then a combined effect of differential abundance and changes in protein complexes. And it again needs diving in deeper to validate all complex changes that you require. And I think since colleagues have come up with more fine-grained and advanced analysis strategies. That said, this is what we could get from this analysis, just an overview of how this looks. Practically, here's the OD traces of the two conditions, triplicates, over the time in these SEC runs, and I would argue they overlapped quite nicely. And then when measuring these 390 runs, 65 fractions in triplicate for the two conditions, and looking at intensity correlation, Pearson correlation here, I think we did a good job. And that is to say, us, Sabine Ammon and Ludovic Scheer and I, who were doing that part of the work. So that went well. And now we get these signals out. We have interesting scenarios where, let's say, on top, we have SEC stable proteins where we really don't think that is a real change. But below, we have some examples where we really think they move around. And one, let's say, positive control was CDK1, which we know should be assembled with cyclin B1. And we saw it moving to an assembled state, so that was good. And now looking into the paper figures, indeed, we could see when overlaying the two that, yes, that's the complex of the two, and we're fairly confident that the perturbation system was, perturbation experiment was complete enough. And it looks good and we can find interesting other things in this data set as well. Good. And one of the other interesting things and a core domain of knowledge of the Kutai lab at ETH, who were the collaborators here and of tremendous help diving into the data and finding interesting examples, was the nuclear pore complex, where we could see the disassembly, the previously described disassembly into subcomplexes during mitosis. So these signals only pop up in the dotted lines here in mitosis versus the solid lines. That's an interface. And that's something where we could then dive in deeper. And Mario spotted a combination of subunits which shouldn't be there according to textbook knowledge. And we dove into that and realized that indeed there is a different composition and a intermediate between, which involves components of both the central channel and the inner ring complex that gets stored away during mitosis. So it doesn't fall apart as much as people thought, but there seems to be an additional complex variant there. And now again, I think people can appreciate how important the nuclear pore complex and its reassembly is for the pace and speed of mitosis, and how important mitosis is also for cancer. So again, I think by having identified new modalities and combinations and also surfaces and interaction interfaces that could be targeted to modulate the pace of reassembly or disassembly, well, probably reassembly, I think we have put a groundstone there for potentially interesting development of interaction targeting cell cycle and specifically nuclear pore complex reassembly inhibitors in the future. We validated this with 2 different ways, but I'm running a bit low on time, so let's not talk about that too much, but rather highlight again that we have invested tremendous time into development of this viewer app. And I think here Max Frank needs to be shouted out, if that's the right way to say it. Sorry, I'm not a native English speaker. And he did a tremendous job here implementing very cool point and click interactive data interrogation options. And I have since played with that as well, learned a lot from him in the process. So I really urge you, if you're doing cell cycle studies, check out this viewer app. There's instructions on page 1. If not, feel free to shoot me an email. I'm always happy to show people how to use it. Should still be online. If not, send me a mail. Cool. And now from this data set, additional analysis were performed, where really the acknowledging the shortcomings of the analysis approach that we had available to date. Others have started developing alternative analysis approaches, notably George Rosenberger, who did an interaction-centric targeted analysis via SECAT, the SEC analysis toolkit, which was published 2020. And that gives a more systematic interaction level view of the data. But also Andrea Fossati and Chen Li must be mentioned, who have done a tremendous job implementing yet another machine learning-based approach to the analysis, and also optimizing the wet lab procedures to get where, let's say, Team Collins is today. So with that, an intermediate thank you, interim, to the team in Zurich, and especially Isa, George, Robin, Max, who have been really central here, also Audrey van Drogen and Ben in this project, and Rudi Ebersold and Matthias Steiger, who were the supervisors here. And with that, I want to just touch on shortly the speed up achievable in the whole process using the EvoSep1 system. And we'll throw this slide at you with some numbers. So this now compares what we can get out of such an experiment when investing 10 times less time, roughly, right? So let's look through the rows. We have the number of protein complex signals at a 5% error rate suggested by our target decoy error model. The number of proteins and the number of protein elution features that we can detect from the protein-centric analysis, and the number of peptides remaining after applying this consecutive ID stretch length filter to not work with anything sporadic. We see that from the initial study, that's the first column, that's MSB 2019, we had around 620 signals, around 4,093 proteins with an SEC feature. In total, we had 4,900, but for some of them, there was just, the algorithm was never sure where it exactly eludes. But just so you know why that number is lower. And importantly, we have 58,000 peptides, and this was done with 120-minute nano LC gradient on an exigent LC, and we could crunch through 7 fractions a day. So that made, for one replicate of 80 fractions, you can do the math, that made 12 days like net acquisition time. And then the best possible speed up we could achieve then for the differential study in HeLa, we had 1 hour gradients on a nano LC that made 12 fractions per day, and measured on the 6600. Those are the results we got. We had a small attrition rate on the protein complex level, likely due to reducing the quantitative accuracy by shortening the gradient. But in principle, it stayed intact. The signal didn't collapse. But now work done in parallel or slightly after I left the lab by Peng Xu, I believe, and also then analyzed with Isabel, was the 21-minute gradient on the Ivosep, and 60 fractions per day, that is, so tremendously faster. And the numbers look exquisite. I mean, they looked really good. And I couldn't believe it, but then I had to believe it. And I think that's one of the building blocks why I work where I work now, because this is really striking. Good, but that work was done by Ben and Peng and Isa and then others as well. So I'm not going to dive into that too much. I'm just going to give a little teaser of the work that was done thereafter then by Charlotte Nicot, Claudia Mitelli, and Isabel, then also especially developing more analysis options, which is this rapid complex-centric quantitative profiling of the monocyte macrophage axis. The preprint is out, new version recently, so please check it out. It involves 3 different conditions, and I'm not gonna try and explain the biology here, that, which resulted in 9 SEC runs. So a lot of fractions, 64 fractions per run, an optimized digest procedure, which I believe Andrea worked towards for his study, and then regular swath analysis on the 6600 with an Ivosep LC. All right, but I hope, you can all take a look at that, and I'm hopeful that we can have another discussion where Ben can, or someone can explain in more detail what was done there. So now I want to explain what the Ivosep box does and what it is, and to understand what goes into this box in terms of knowledge. I think it's also interesting to look at the trajectory of, and now I'm putting on my Ivosep hat, of us as a company and where Ivosep came from. And the core team really has been innovators in the field of spectrometry sources, MS in general, and then LC systems in particular, since the foundation of Proxion and making the easy NLCs that many of us know. And also then the easy spray columns and sources after Proxion was acquired by Thermo. Then transitioning to Bruker, where the nano elute was a key product of this innovative team. And then in 2017 or so, or 16, founding Ivosep and really putting together a rather new and transformative concept of how to do liquid chromatography. And I'm going to dive into that now. I'm going to skip over that in the interest of time. Maybe to clarify how this looks, if you haven't seen one yet. So it's this box, it has the buffers out front here, and what you see on top here, that's not the tips for pipetting, that's the Evo tips, and that's basically stage tips. It's proprietary, and you need to use ours, where there is a specific optimized C18 material in the bottom, and that's where you load your peptides. So you don't have vials standing and evaporating, but you have these tips filled with buffer A, water, formic acid above, and also buffer A below. And then they sit there until you analyze them. And the machine picks it up with a little needle that's over here. I'm not sure if you guys can see my pointer. Could someone comment?
Yes, we can see your pointer.
You see it? Okay, cool. Right. Yeah. Otherwise, it's just confusing. Okay, perfect. Thank you, Tiana. Yep. So it comes over, it picks up the sample, then it goes into this inject port here, and then interestingly, we have, and that's the view of the inside, we have low pressure pumps which form the gradient and form a first gradient and elute the peptides off of the C18 material into a sample storage loop. And then a second set of low pressure peptides make a second gradient. And I think I have a graph for that. So that's the cool part. It's done at low pressure, pretty fast, and without much strain on the pumps and system itself. And then there's a fifth pump, if you count it right. Yes, if I count it right. And that's the high pressure pump, which then just pushes that stuff onto the analytical column and it gets refocused. So it's not technically like binding the peptides to the analytical column and then eluting them with the gradient, but it is pre-fractionated peptides eluted out of this tip, put into a loop, followed by another gradient. And then these together get pushed over via the analytical column to the source. So that's, I think, strikingly different from regular chromatography, regular nanoflow chromatography at least. So let's look at this as a scheme. So here you have the four low pressure pumps A, B, C, D. There's the tip here in the middle. And the gradients are illustrated here over the volume of the piston, the composition of buffer B, and the green one is the first gradient. That's the one that is made by A and B and pushed through the tip. And then after this is done, this offset gradient generated by pumps C and D places the second gradient behind the sample into the loop. So the valve is switched differently than in this picture, right? In this picture, it's currently pushing out sample 1 onto the analytical column into the mass spec. But when the sample is eluted into the loop, obviously the valve is in the, what would it be, 1/6 position or something like that, right? So that gets pushed into the loop, and then the sample gets pushed out over the analytical column. And from this offset pre-fractionation from the first gradient, we reside at a sharpened picture as they arrive at the source, at the electrospray, but it's really a refocusing system. All right, and that's the chromatography down here, an example picture. And of course, one can do different things with it. I'll skip over that one. I think one cool part that I want to touch on is the fact that this Evo tip factually is a desalting device, right? It's a C18 column. And while we typically would load clean samples that have been cleaned up before, theoretically, if a digest is rather clean, it can spin out all the crap and then load some of the supernatant directly and at least be adventurous to try out a direct cleanup and directly separate that after cleaning it up on the Evo tip, and really skip yourself a lot of the pain with the drying down and the vials and all that stuff. So yeah, that's, I think, something nice and something I did in the past, and that really made my life in the lab easier and brought me to more data faster. Speaking of faster, this is also what EvoSet provides. So the system can do different samples per day. That's how it's termed. Starting from 30 samples per day, the deepest proteome coverage going down to 300 or up to 300 samples per day when you need really high throughput. And that then relates to specific gradient lengths. So what is the time of actual gradient arriving at the column? And what is the cycle time realized by this method if you add the overhead of the low pressure pumps regenerating the system and doing preparing for the next sample, which are arguably very short. So you achieve a very low cycle time and just can crunch through many samples. And how the system then achieves this short measurement time really is by pushing this gradient through the analytical column faster at a higher flow rate. So we have low flow rates at 30 samples per day, 500 nanoliter per minute, and a maximum of 4 microliter per minute in the 300 samples per day. And then there is specific LC column choices, and we sell these columns that match and are well characterized for the respective methods, with a set of endurance columns that one can just use at room temperature, or a set of performance columns with smaller particle size that give even better chromatography and results, which then need to be used with a heater device. And for anyone who got interested and wants to read up more, I can direct you to our amazing website, where there is so much information, applications that have been demonstrated both by customers and collaborators. And there's also a nice literature room where you can search for certain application types or keywords. And we have a short list of interesting literature there for you. And I also encourage you, even though timing might be difficult from China, to join the webinars ever since COVID, and probably also before. An excellent series of webinars covering different topics has been organized and is hosted by us. So I really encourage you to check it out, read through it, also look at our videos of sample prep. If you have hesitations, tip my sample on a tip, I don't know, look at it, talk to people who have the system. I can only recommend it. And that is Moritz speaking, and not only Josef speaking. Okay, and with that, I made it to the end, and look forward to a nice discussion. And thanks a lot to everyone involved in the studies I presented. It was real teamwork, and everyone at Evosep also for giving me the opportunity to now, during a visit elsewhere, jump out and do this. And then I look forward to your questions or the discussion we can have.
Thank you, Dr. Husel, for your exciting talk. We have just had quite a journey with you. We can see since 2017, your doctorate dissertation that you mentioned first time, I think the SEC, SWAS, MS, it has come a long way, as we can see, to be the unbelievably powerful tool that it is today, and one of the best performance strategies for complex centric profiling. So Dr. Guo, would you please start leading the panel discussion so we can, Yeah,
thank you. I have a quick question. So you have a webinar for Evosep. Do you have a recording for this webinar? I know this is not a favorable time for the Chinese audience.
Yes, there's typically the webinars are typically made available offline, and you can rewatch them at a later time. So you just sign up, and then typically there's a link sent, and you can watch it later. And yeah, I encourage to revisit the old ones because they're really good.
Okay, a technical question. So I understand you now can inject 30 to 300 injections every day using the Evosep. But how about the lifetime of the column? This will be a huge load for the column. Normally a column can inject for 1,000 times or 2,000 times. Then does this mean the column will be, the lifetime will be shorter, or is similar to what we used in the
I mean, maybe, yes. So I can comment that in my experience prior to joining Evosep, when working with the Evosep during my postdoc in Sweden, but also then at the company now, I'm met with high robustness and really stable analytical column back pressures owed to how the system is designed. So the gradient that goes over the tip only elutes up to, say, 50%. Maybe I'm getting the number not exactly right, but the high organic and often contaminant-containing, lipid-containing fraction of what a proteomics sample can sometimes contain, that tends to clog up your nano columns when you just do a regular wash after injecting your full sample onto that column and wash with high organic. Your lipids accumulate and your back pressure gradually increases. Depending on sample quality, I've seen nano columns and nanoflow systems operated without a pre-column clog up after 20 runs when the samples are tricky. And because of the way the EvoSAP is built and the partial elution of the tip and the fact that each tip is disposable, they are not reusable. That's a single-use item. It also gets deformed as the injection happens, so it's also not encouraged to reuse. Therefore, you throw away all the dirt that there might be in excess of the interesting peptides, and you elute only the peptides, and then the junk goes into the waste where it belongs and not into your analytical column and source and mass spectrometer.
So normally, how long can the column last?
I think, I mean, the numbers that you outlined to me sounded like EvoSAP numbers. They didn't sound like nanoflow numbers to me, like 1,000. It's hard to guarantee a certain number, but certainly longer than on the alternative. But I can't give you a guaranteed. I don't think there's a warranty number, as in, you know, if the column dies before 200 injections, it will be replaced. I don't think so, but I'm also not aware of that. So I might need to ask for that, and I can shoot you an email with the answer once I have consulted with my colleagues.
Another technical question. This EvoSAP, you need to use a disposable tips. And just like you mentioned, you do not reuse it because you want to remove the dirty stuff to save the column. Is there any other reason, or let it put another way? Since it's so expensive, is it possible to reuse them for some of the samples? I think to answer that clearly, no. Like you can't reuse them. If you do, you technically void system warranty, and the reason is because, as you will see if you follow closely when you see one of the systems, the injection cycle, you will see how firm, to seal it off properly, the tip is pushed into the needle seat, and the ceramic needle really kind of destroys the tip, right? Yes. And then in a second injection cycle, it won't be tight anymore, and there's nothing we can guarantee, and all the validations and all the guarantees and all the performance really is built around this disposable concept. That said, I think it's important to note that if you load your digest directly on a tip, or if you have your sample and load it on the tip and store it up to 2-3 weeks in the fridge before measuring it, you technically, okay, that's two scenarios, right? In the one scenario, you don't need the C18 cleanup column anymore, right? Your Oasis cartridge or whatnot. I shouldn't name specific vendors, but whatever you use to clean up your samples, you might be able to skip that. And if you look at the price at those, you will realize that the price of one Evo chip is about the same. It should be. If not, let me know, and I can do some research why in a particular market it would be more expensive. So likely you are spending the same money, but saving time. That's my best triangulation of that scenario, and why I think that cost in the bigger scheme of things, and especially the cost of nano columns that clogged up and need to be replaced more frequently, you will save that quickly. So that's my opinion. I have a question for the complex study. So you just now you showed a few very nice examples. Do you see some peaks, and this peak move to the other P group, means there's a disappear or join another complex? But have you checked, is there any stoichiometry difference? Let's say the ratio of certain protein, they flip, they are both there, but the ratio change. Is there any some example like this?
Yes, yeah, there is examples like that, but we haven't, or at least to my knowledge, there hasn't been a good computational solution. Maybe in George's Sekat tool that has the most sophisticated assessment of this relative intensity changes that would indicate stoichiometry change to look at that systematically. That said, one can always look at it manually, and if in the two conditions you quantify the proteins on the same set of peptides, which disclaimer and word of caution, many tools don't do, and just select whatever peptides are there in each sample. And if the visibility pattern or the peptide pattern or the set of peptides obtainable in the one context versus the other is different, one needs to make sure to quantify, in my opinion at least, one needs to make sure to quantify on the same set of peptides across both conditions, and then, you know, eliminates all the factors of flyability and so on, to then really be able to say, yes, there was a stoichiometry change here. Plus, I think the replicates have to be there to actually have some sort of statistical scoring and confidence in making the statement whether this difference is truly something that is worth following up on, or within the random effects that are in any such data set.
Yeah. Hey, Dean, can you mute yourself? Yeah, there's some other Longguang and Mengge. Zhiwei, do you have questions?
I have some questions. You have made a great step to identify the protein complex system-wide and quickly, but I think that there are still a lot of protein complex that are undetectable, even we use the advanced technology. So this kind of protein complex may be important or biological meaningful. So I want to know what do you think is the most important factor that brought the technology from a deeper detection for protein complex?
Yes, that's an excellent question. So what limits the technology still, and maybe the approach as a whole, is the keeping the complexes intact, right? Because I think what one needs to appreciate about this biological system is that we don't have complex, non-complex. We don't have like interacting, non-interacting. It's not a 0 and 1 situation. And no single method that we use to study these complexes or these protein-protein interactions will give you the same identical picture, because it will affect the state of these interactions in a different way depending on the experimental parameters. So every method can only offer one angle onto the system, and we should really use multiple methods in parallel to understand the dynamics and also the spectrum and the continuum of interaction affinities, and also turnover rates, how quickly a certain complex forms and falls apart. Now for SEC and SEC SWOT, as in the workflow we worked out in Zurich, and that applies to, I think, some of the previous studies from Christensen, Foster, as well as Emily. But let's focus on ours and the SEC approach, is that you only carve out and see the set of complexes that can be obtained from the cell with that detergent, with that like freeze-thaw lysis that we do. Then you might, if you do a sonication step, you might kick some apart while you might get others out, right? So as soon as you change the wet lab protocol, your picture and set of complexes that you get will change. And in this scenario, it's just a set of experiments where it's so important to control the experimental parameters as tightly as you have never controlled any experimental parameters, you know, and keep it cold and work so accurately to make sure your replicates line up, because otherwise this quick falling apart of complexes or the ratio between an assembled and non-assembled, if that is an effect of the protocol and not a very strong biological truth that we are looking at, needs to be taken care of. And yeah, it really only gives us one view, and I would say a view towards the little bit more stable set of complexes as it is implemented now, without any cross-linking or complex stabilization, but just lysing and keeping it cold and quickly getting them onto the column. Long answer. I hope that answered your question, or if I can elaborate more if you want.
I have some detailed questions. Maybe I need the first question is about the page 46, the summary table. Did you get the complex number from only one experiment?
Yes, yeah. That's a single experiment with HeLa cells run through the CC profiler pipeline, right? So this is before any differentials, also not with the goal to do differentials. This is really just one cell line sample processed through the pipeline and run through CC profiler with the same set of complex hypotheses as the other data sets.
But actually, I read your paper, the Nature protocol or the on the bioRxiv, you always use 3 replicates for one samples. Why? You need to keep the complex traces more robust, or other reasons?
So in the case of the differential studies, and that includes the HeLa cell cycle study, Cell Systems 2020, and the current preprint from Isabel, Charlotte, and Claudia and Ben, that all is differential. And the first, let's say, differential testing pipeline that we developed was really based on this, looking at the protein abundance in a certain range along the SCC. And in order to do any useful statistics there, you need, or we wanted to have at least 3 replicates on either side of the comparison. So that is why in all differential studies, we said 3 is the lowest number we are happy with. And in the case of the initial study in the molecular systems biology, HEC 293, let's say method development study, we used initially a single replicate, and then after finding the interesting complexes, we repeated the experiment for that range. So I would say it's a 1.5 replicate experiment, which arguably isn't ideal. But I think given that we have all this information, how the things elude from the SCC column, and tight conformance of this partial replicate 2 with what we've seen in the same range in replicate 1, and we have that in the supplemental if you're interested to scrutinize that side by side, that's where it is. And yeah, I think with this additional information of SCC and this peak shape, I'm not a single replicate friend, but that's what we could do back then. And I think we still have found very interesting biology. Going forward, I would, of course, recommend doing 3 replicates and in combination with higher throughput chromatography, and or, I mean, people are also exploring like labeling techniques to multiplex, and the original Tristans and Foster study was done with SILEC, right, to combine the different conditions into the same SCC experiment, which is a powerful approach to remove any variability from SCC. Also a viable and interesting approach has been done. And going forward, I would really recommend at least 2, better 3 replicates side by side, especially so if you want to compare different conditions.
And another question is...
Sorry, Murray. So how reproducible is the SEC chromatography and the fractionation? Even for the nanos and LC, there have some variability. You run 3 injections, and over time. And so the SEC, you ran, the protein complex. Just now you mentioned the stability of the protein complex. When you do lysis, tissue or cell lysis extraction, there's a lot of technical issues, temperature, whatever.
Yes. Yeah, that's, I think, what I alluded to before, that it just needs to be super tightly controlled, as I believe from the moment when you, and that's a somewhat theoretical consideration, but from the moment you pop open that cell in a lysis buffer, the situation changes dramatically and the complexes start being unhappy, falling apart. No matter how nice your buffer, it's just a mere, physical chemistry effect and thermodynamics effect that you dilute them out, and then everything that has a quick exchange rate, high off rate, K on, K off in a certain interaction. I mean, the same interaction or two interactions with the same KD can have significantly different K on and K off rates. So, there's an additional dimension to interactions that we don't fully acknowledge in our regular, let's say, mass spectrometristness. And so this needs to be controlled very tightly. And I've seen replicates look very different, and it was then tracked down to differences in, for example, the time between lysis and when it went onto the column, or the temperature not being controlled consistently. Of course, lysis itself also becomes super critical. And the more this can be automated and put into a standardized platform, like I believe, some developments are on the way in that direction. I think that's a good idea, right? To put it into some sort of device that does it, then you already remove a lot of variability on that level. So I think that's an excellent route forward to control these critical early experimental parameters in this workflow more tightly.
Yeah, Munga, go ahead.
And the final question is the complex number from, did you got the complex number from multi-complex databases, such as all of the complex?
So those numbers, I think in that slide that I showed, and that's work done by the later parts, right, by Peng and Ben and Isabel. I think the goal was just to compare with the same complex set across the different methods, if it goes down or up. And I think what was used was the quorum database at the current version back then. So it did not integrate string and bioplex, which was integrated for the big analysis for the MSB paper.
So maybe the overlap.
Another question. Have you tried the FFP samples? A lot of clinical samples are FFP samples.
Yeah, no, I haven't worked on clinical samples at all. I really left after the HeLa CCL2. And of course, that is a tricky thing. And FFPE fixation, I would think, has profound effects on protein complexes. And once everything is locked into place by high concentration in tissue, cross-linking like that, I think there's little chance to get really meaningful differential biology in terms of higher order proteome organization out of such samples, to be quite frank. Like digesting them down, getting the peptides and getting a regular proteome pattern from those samples, yes, that has been shown and proven, and people have done really good progress towards getting deep proteomes out of it. But for the complexes, I would not be very happy to try and optimize that, because you have cross-linked in a super high concentration, and you have everything is next to each other, even if they don't technically interact. But then you cross-link them, and then what are you going to do about that later?
Have you detected complexes which are not in the databases?
So with our analysis approach, this is inherently not possible, right? Because we say, in order to overcome the problem of too little resolution and difference between all the different complexes in only one chromatographic dimension, we need to come in with this targeted information, targeted analysis using external reference information but that inherently limits us from detecting new complexes. If new complexes were sought after, and I think there will be new ones, some, but they need to be very carefully validated. I would really recommend looking into work from Andrew Emili and HuB et al. Also Van et al. 2015 Nature paper, where they did this with super many chromatographic fractionations and different species, and then doing orthology mapping of the genes. An amazing work, and an algorithm like capable and geared towards from these different species and mapping the orthologs across species and the coevolution in multiple different chromatographic dimensions to really suggest and detect complexes that have never been observed before. And they make an assessment of like how much of the complexes they report have no reference in public databases. So that I would recommend looking both at their software and at their approach in general. It's a really great piece of work.
So did they detect the complex by mass spec or other methods?
It's also mass spec, but really focused on going. So initially, the HuB 2012, which I outlined, that was a cell line or 2 cell lines, and then 3 dimensions, and then like kickstarting the first iteration of that algorithm to find complexes, and then the newer ones, 2015, that's then across different trees of life, or the tree of life, different species, and it's all done with mass spec. But different mass specs, I mean, so many fractions, you can't believe it. And I think they just used any mass spec that wasn't doing anything else. I don't know. If you look at the data set and supplement and the methods, you will see that this, has been done in so many different sites, and it must have been a huge effort coordinating all that. So hat off for coordinating that project.
We should check that paper. Yeah.
Yeah, I can send it. Yeah.
So Wenguang, do you have any questions?
Yes. Hi, Morris. Thanks for the nice talk with lots of information and details as always. So thanks Westlake Laboratory and Tiannan for this webinar and the invitation. So I'm just curious about your EvaSAP and the ProteinComplex, and also the computational workflow. It looks like the DIAN seems to be a perfect case to fit for the data analysis, where it was which was originally designed for short gradient LC. So what do you think? Did you try in the EvaSAP company to use DIAN workflow, or you are also still using Spectronaut or open source, or any comments?
Yeah. So on DIA analysis in general, I guess, it's just amazing what has been contributed to the field over the last few years after it got more popular. And DIAN is one of these very important landmarks. And I do use it a lot. I used it a lot in Lund. We are using it a lot now. So it's really a core workhorse for our rapid interpretation of DIA data. And yes, especially in combination with the short gradients of the EvoSAP1, both the software and our instrument excel and work together nicely. Also because, I mean, as you make the gradients shorter, you get more interference, and I think DIAN is really good at removing interfering transitions and refocusing the analysis after an initial pass onto the transitions that are less interfered, and that's perfect for short gradients. And I think when we look at the important work out of the Oralza lab, and when DIAN was developed by Vadim Demichev and his colleagues, was also a short gradient and short measurement application. So that inherently is highly compatible and a strong analysis platform for the EvoSAP data, yes.
So and then a follow-up question would be the library. So you mentioned a lot about the library, and any more comments for example, using DIAN, you use direct DIA, or you're still trying to build a library based on DDA data? Do you have any experience or comments related with that?
Yes, yeah, happy to share. And I think that's a long topic. We can maybe also have an offline meeting, catch up and go into the details. I would be actually happy to catch up with you after this. Maybe it's good to see you again after so many years.
Yeah. It seems that you give up WeChat. So I sent some message on.
I had to for data reasons. I just yeah. Anyway, so the Diane library, I think the library free is an excellent option in Diane, in particular in combination with the match between runs feature available. The match between runs really, when looking into it in more detail, doesn't do anything like match between runs in MaxQuant DDA, like MS1 feature matching, but rather makes a new library from the initial path analysis, then refines that library and targets the library more towards the set of peptides and proteins contained in your sample set. And that's really powerful and helps a lot. And with that, I think with a library-free analysis relatively to a optimal sample-specific, project-specific library, you are getting to 85–90% in terms of the numbers and also a really good overlap. So I think that's a strong platform, but it still for the first pass requires quite some computational resources because you search this whole whatever 4 million precursor or even more if you want to consider modifications and whatnot. So it requires the resources, and due to the huge search space, it's not super sensitive, meaning that if you start the analysis with, let's say, the pan-human combined assay library from the Eversole Lab 2014 George's paper, you have a chance to get a bit deeper because you start out with a more confined set of proteins, and this, let's say, statistical error control limiting the sensitivity of your validation of P groups in the first pass analysis is less troubled. But yeah, it's a long topic. So what I now do and like to do, and I'm happy to share, is like using for typical analysis where I don't have reason to believe that there's other proteins in there than those in George's library. I use George's library, and whenever it's a different mass spectrometer than it was acquired on, I use Diane to, and the Diane included prediction model to predict the retention time and fragmentation and also ion mobility of that set of precursors. And then that's a sweet spot for me where I get good results.
So that would be my final question about the prediction or generation of in silico library. You always use Diane to predict the library. Did you try other tools?
We did try other tools, and in particular, like a very talented PhD student in Lund, who I had the joy of collaborating with and playing with tools, and yeah, tried different things there. And also, yeah, the computational side of the group in Lund has been trying different ones. That said, yeah, the ease of use is, I think, very good on the Diane side. Of course, also other alternatives like ProZit offer good predictions that are compatible with the DIA workflows, and that has been demonstrated by the Küster and Wilhelm labs in Munich, but also yeah, by the McCoss lab, and yeah, when they did the refinement of the libraries based on gas phase fractionation, searching that with like searching gas phase fractionated DIA data with a big predicted library, and then making a new one from that, and then throwing that onto a non-fractionated single shot DIA run. Pretty cool paper as well.
So there's a question from the audience. Hugo, do you want to speak by yourself or?
Yeah, I can. Hello. So I'm sorry I arrived a little bit late to the talk, but I'm working also in complex and profiling, and I work with Christoph Lenz's lab in Germany also. I want to ask you, so you developed CC Profiler in your group to study protein complexes with your workflow, and then you published something else later on And I want to ask you which are the possibilities to detect, for example, protein alteration within the same protein complex, but compare between conditions, between two different conditions. If there is some bioinformatic workflow to follow that you develop.
So you mean like also stoichiometry change, that in a certain complex P group, one of the subunits becomes less or more abundant in the different condition? Is that it?
Yeah, that one thing could be this alteration in stoichiometry, but also changes in the protein profile within the same protein complex, maybe.
Okay.
How meaningful would be that also?
Yeah, good question. So I think it's one thing to look at just the, let's say, area inside that P group, if it goes up and down, and that is more simple. I think it's part of the SECAT profile and SECAT software. And I think there's also a module in that in the preprint that comes out, like the complex-centric analysis. I'm not sure to which extent like outlier proteins that change their abundance or their relative contribution to the P group between conditions, to which extent this is explored. But I really hope you can check out the preprint there and dive into the supplement also. Should be out soon, hopefully. And perhaps we can also have an additional discussion, or you can contact, the lead author there, Isabel. She's the one who worked out the downstream analysis on the complex-centric level. So perhaps she can have a nice comment on that as well, or even hints on usage. Now, when it comes to the shape of the protein inside that P group, I think that is still very tricky. And given the uncertainties of, let's say, protein quantification and the workflow, I wouldn't be sure, at which point I would consider this significant or not. And there really a lot of replicates would be required for me, at least, to be convinced that this is an indication that could lead me towards expensive and timely experiments to try and validate any hypothesis I might be able to derive from a different peak shape inside the P group. I hope that answers your question.
Yeah,
I'm not sure.
Completely. Thank you.
Okay, welcome. Thanks for the question.
Hi, Morris. I have a last question. So I know the EvaSep you're talking about protein complex, but EvaSep has a huge potential, I think, in clinical applications, because now you are analyzing really large number of sample in a very short time. For clinical application, this instrument is for research only. So at the EvaSep, do you have plan to, let's say, move this forward to be a medical device in the future? What's your view or outlook in this perspective?
Yes, that's a very interesting and important point. And yes, the instrument is very capable for larger cohorts, and I think that's really the premise of the company and let's say a hope or a long-term development goal. But I am honestly not aware of where this exactly is headed and where we stand in the process. So it's definitely geared towards clinical proteomics. Whether it will make it into the medical device section or not, that I don't dare to extrapolate, but it's for sure today already for larger clinical studies and let's say the basic research happening at the clinic using clinical samples, a very good solution. And ideally, of course, we would really like to see the instrument in the clinics being used as a diagnostic, as part of a diagnostic procedure or whatnot. But that is a very complicated regulatory space, and I frankly am not the one who makes any of those decisions in the company. I'm happy to inquire about that internally and give you the feedback I can give you, but I don't really know, to be honest.
That's cool.
But,
so how about, you mentioned you have several programs from 30 samples to 300 samples every day. This is quite weird, you know, you do not allow users to change. Do you have plan to make more flexibility for the users?
Yes, so yes, I agree. It seems firstly to a mass spectrometrist odd to be limited to the set of methods, but let me promise you they are super highly optimized, and anything you might want to try with that gradient, and also the fact that it is not a conventional gradient. It doesn't work as we think it works, and it's not AB running over a column, right? It's a gradient formed in a loop with a certain flow profile, with a certain number of valves and a certain length transfer capillary involved. That's Pandora's box to open that up. And if the goal is standardization and robustness and also improving reproducibility between labs and ease of use, I personally think that the decision the company made when designing the product was the right one to limit the methods and have very clearly specified methods that can be selected from and that serve most purposes. That said, in addition to the methods I showed you on the slide, there's the EvoSep Plus methods, which then address specific needs of collaborators, key opinion leaders, people who have a specific application that they really want to make work on our instrument. And that is something where we always have an open ear and can discuss, and I can always promote that internally and maybe even think about some sort of collaboration to push for the development of a certain method that would serve a reasonable user base to then, you know, really allocate resources to make that happen. I don't make those decisions, but this has been done in the past for certain applications. One user desired such method as the extended method to run 1.5 hours to really dig deeper into proteomes with the performance column in that combination, a bit lower flow rate, already then also bumping sensitivity or a little bit more up, so it's 220 nanoliter per minute, or then the whisper methods, which really target the single cell and super low input sample range, and also in combination with an extended range of columns, as we have recently demonstrated for single cell proteomics, where it can excel and go beyond 2000 proteins quantified from one single cell without any carrier channel, label-free DIA. Man, I didn't believe it at first. That's what it is. So the range of methods is extending, and it's of course a balancing act, I presume, you know, between having too many methods out there and being standardized. But perhaps there is still some space that I'm happy to inquire. If you have something specific in mind, maybe we can also have an offline meeting afterwards.
Yeah, cool. So what's the future plan for EvoSep system?
Well, I think the clinics you raised as a target is clearly a plan in the roadmap of the company. Of course, I think it's no secret to say that we are not yet in all markets equally, and more labs and experimentalists can benefit from higher throughput and less sample prep work that we can offer, and that is clearly the interim strategy to really go out there and showcase new types of applications on the instrument and show what it can do in all different types of domains that are compatible with the build type of the system. So for example, I just recently, my first real project in the company was trying out peptide mapping of biologics and characterizing how good we can do that and how good the retention time, reproducibility, area reproducibility is on the system, qualifying the system for things like QC in pharma companies, be it early on in pilot studies or downstream, even in parallel to production and QCing biologics as they get produced for shipping. So there's a lot of spaces where we are not yet and where we can still go, and we are always open for suggestions and ideas, and yeah, to discuss.
Yeah, it's a very smart system. I look forward to collaborating with you in the future. So time goes fast. It's now already almost 1 hour 15 minutes. It's over our schedule. So thanks for lots for your effort. I heard a very nice talk. Very good, actually very good training of the mass spec strategies, EDA, TIA, SRM, and also a very nice talk about the complexes. I think this is a huge field forward. We now understand protein not in a singular format, but also in a complex in context. But I think it's just only a start. You can only detect 500 complexes right now. In the future, with a semi or Bruker instrument with the EVA-SAP, I think we can dive much deeper and get more replicas, more complexes, and more species, more conditions, and get more information. Thanks a lot for your time, and thanks for the support from the audience. And I think we have to end now. And thanks a lot. And see you next time.
Yeah, thank you so much, Yanan, and everyone, the panel, and all the listeners. Thanks, and have a good day.
Yeah, have a good day. Bye-bye.
Bye.
Bye.